Okay, so today we are looking at the structure and function of DNA. You already looked a little bit at its discovery, and hopefully some of this will kind of resonate with what you've already seen from the video in the play posit. So for your assignment for today, I just need you to go ahead, get a piece of paper, and as I am going through, I want you to define or describe each of these terms. And I do want you to keep it specific to how I'm describing it here, so don't go off and Google them and get way too many details. Keep it short, simple, to the point, and that's fine. So two questions that I want you to be able to answer are the two functions of DNA, and then what does the central dogma say? In terms of vocabulary terms, again, you're not needing to give me this like huge description. I want you to write down what a nucleotide is, what are the various parts of a nucleotide in general and specifically relating to DNA. What do I mean by backbone, by strand, by base pairs? Both specifically, what are the rules of base pairing and what does base pairing mean? The term antiparallel, coiling, and then finally chromosome. So if you just want to go ahead and write those down on paper and then upload them into the Schoology assignment for today, you'll be in good shape. All right, so let's go ahead by starting off and looking at the function of DNA. There's really, as we've mentioned, going to be two, and this is the foundation of the rest of the unit. So the genetics unit is going to really be broken into two parts. In the first, we're looking at the aspect of DNA that allows it to carry instructions. And with those instructions, we're going to want to understand how that makes you you. So it's not that the DNA per se is what makes you you, it's that they're building proteins. So the instructions for making proteins are in the DNA, and proteins have tons and tons of different functions, enzymes and structures. So really it's the proteins that make you different from everybody else. But what determines what those proteins look like is in the instructions of the DNA. So we're going to start this unit off by looking at how DNA is read. The second part of the unit is all about how that information then gets passed on to offspring. And that's kind of the theoretical genetics that you're probably more familiar with, that you're probably thinking about when I talk about genetics, things like Punnett squares and so forth. And so DNA must not only be read, but has to be copied. In a lot of ways, when I talk about DNA, I tend to refer to it kind of in book terms because of how useful that analogy really is. Okay, moving on to the central dogma. This was named by Watson and Crick, and the idea of the central dogma is that a theory is science's kind of gold standard, right? It is supported by evidence and well accepted and explains a whole bunch of natural phenomenon. And Watson and Crick thought, this is such an integral idea in biology that it's not just a theory. Theories are amazing, but this is the thing that changes how we think about biology. So the central dogma states that DNA is going to be used to code for something called RNA, a molecule that we'll look at here shortly. And then that RNA is what is then used to build proteins. If we're thinking virology and viruses and the replication cycle, this is where things get a little bit interesting. So I'll be making some videos on those shortly. So there's your central dogma, but it all comes down to DNA. And what is DNA? Deoxyribonucleic acid. If we're talking nucleic acid, it's worth mentioning each of our macromolecules. I know this is a really old topic from like 8 years ago, it feels like, but let's go ahead and review what our macromolecules are and what they're made out of. So remember that carbs are made out of monosaccharides. Lipids, like fats, oils, and waxes, are made out of fatty acids and glycerol. That our proteins are made of amino acids, which we will come back to quite a bit in this unit. And then finally, nucleic acids are made out of nucleotides. For each of these, remember that we've got small units, our monomers, that get polymerized or strung together to make the macromolecule. So the monomer here is the nucleotide, and when a whole bunch of nucleotides are strung together, you end up with a nucleic acid, in this case deoxyribonucleic acid, or DNA. So let's go ahead and take a look at what nucleotides are. There's going to be three components. There's the phosphate group, there is a 5-carbon sugar, and then there is something called the nitrogenous base. You might see it as N-base or nitrogen base or just base. And these three components are all covalently bonded together, so they're really strong. They're just as strong as anything within each of those three components. So the phosphate group, the 5-carbon sugar, and the nitrogenous base are going to be found in all nucleotides. And there are variations of each of these things. So for example, one example of a 5-carbon sugar is the sugar ribose. Remember that ose, and we're thinking carbohydrates. But in the case of DNA, we're not using ribose. We're going to deoxy this, or remove an oxygen, and that's going to make something really similar to ribose, but not quite, which is deoxyribose. So now we understand why it's called deoxyribonucleic acid. And then the last component here that we haven't yet talked about are the nitrogenous bases, which can be these big things or these little things. You don't need to memorize which is which, but we will see that it's pretty important here in a second. So there are 4 nitrogenous bases that you're going to find in DNA. You're going to need to know them. They're adenine, thymine, guanine, and cytosine. Be able to say them, but to keep things easy, we typically refer to them just by the first letter. So A, T, G, and C. All right, let's take a look at how these individual monomers are going to then come together to form DNA. So what we have is a strand of DNA that is formed right here. We've got a single nucleotide here, here, and here. And so we've got the phosphate group and the 5-carbon sugars alternating in what we're going to call the backbone, which is just the piece that isn't the nitrogenous base. So the sugar and the phosphate, which alternate and make up the back piece, or the spiraling piece of the DNA. And remember that the movie that you watched yesterday was called The Double Helix. The reason for that is because of the fact that it is 2 strands. And so these 2 strands are going to run opposite of each other, anti-parallel. So the 2 strands run opposite. So you'll see here a phosphate group kind of pointing upward, and down here, that same phosphate group is pointing downward, and everything's kind of flipped. Okay. And then in the middle, we've got these nitrogenous bases that face each other, and those are going to be held together with hydrogen bonds. So remember from our unit on chemistry and water that hydrogen bonds are weak intermolecular attractions. In other words, attractions between 2 different molecules, and they're going to be attracted because of a slight positive and a slight negative charge. Okay. So the nitrogenous bases have different areas that are slightly positive, slightly negative, and they're going to attract to each other. And they're going to attract in such a way that A always is going to bind with T. So we call this the rule of base pairing. A always binds with T, adenine with thymine. Easy way to remember that is A's drawn all with straight lines, T's drawn with all straight lines, and then the other set, C's and G's, are also always going to bind. And the reason is because they're different sizes. So you always have to have one small with one big of the nitrogenous bases, right? Remember we saw here that they can be 1 ring or 2. Gotta wait for that. There we go. So the A's and T's, C's and G's, always going to bind together. So let's go ahead and take a look at what DNA looks like, and now take what you're probably thinking in your head as DNA and see how it actually looks here. If I can remember where the web page went. Where did I put it? There it is. Okay. So MoleView, your favorite. Here we've got a double-stranded piece of DNA. Now this is actually a tiny little bit, but we can take a look right here, right? This one red, each of these individual red pieces represent the nitrogenous bases, and then that's bonded to the backbone that kind of spirals down, and this backbone would just be sugar phosphates repeating over and over again. They don't tend to add that detail in because it's always the same. It doesn't change. What matters are these rungs of the ladder, as they're usually called, or the base pairs. In this case, we've got 1, 2, 3, 4, etc. I believe I counted 12 base pairs. To put that in context, you have 3 billion base pairs in your genome. And every single cell of your body, save for some red blood cells, have all of your genome. So your DNA is 3 billion base pairs long, and you have enough of it, or you have 3 billion base pairs in every single cell. So we can imagine that these are like super tiny, okay? And in fact, a really important thing about DNA is the fact that it can coil up. And this coiling is what's going to allow it to condense tight enough that it can fit into the nucleus of each of the cells. Sorry. So we've got the DNA strand, and that's going to wrap around proteins that are kind of holding it into place. And then those proteins kind of wrap around each other, and we get this coiling upon coiling upon coiling until finally we make what is called a chromosome, or one molecule of DNA that's super duper condensed with proteins to hold everything together without it getting tangled. If you've ever heard of like 23andMe, 23andMe is a company that sequences your DNA, and the reason they're called 23andMe is because we have 23 pairs of chromosomes. Fun fact. Anyway, so now moving on, my hope is that you have plenty of time left today, and you're eager to work on some more bio. So I'm going to introduce the project that will be due tomorrow night, which is this. You're going to make a DNA model. And what I want you to include is at least 4 nucleotides. And within each nucleotide, I want the 3 parts, right? So you can clearly tell this is the phosphate, the 5-carbon sugar, and the nitrogenous base. You have everything bonded correctly in terms of the nitrogenous bases facing inwards towards each other, and then the other piece is part of the backbone. I want you to include 2 separate strands, so they should be running anti-parallel, and on each strand you'll need at least 2 nucleotides. Bonus points if you can get a big enough model going that you can really start to see that twist. But I don't want to stress you out. Stick with 4, make your life easy. But instead of just taking a picture, because, well, that's lame, right? If I'm sitting here every day, making videos for you guys, I'm going to want one back from you. So here's what I need. You're going to take your model, and it's okay if you can't like pick it up and move it around. Hey, bud. Speaking of DNA, this little dude, come here, you. This is my model of DNA because he's got my DNA in him, and I have a nice burnt marshmallow. Anyway, you want to go play? Go play. So you'll make a short video of your model, and I want you to just narrate it in such a way that I can tell what the nucleotide is, what the phosphate, 5-carbon sugar, and nitrogenous bases are within each nucleotide, and then also go ahead and show me where the strands are, what is the base pairs. Like I said, it's not something you have to be able to even pick up. So if you want to just make it out of different shapes using paper, that's cool. Play-Doh, that's fine. If you want to get creative and go crazy, have at it. But what's really cool about this is when I say use household materials, I really mean it, and none of you can go out to the craft store because it's closed because of the COVID. So you'll have to get creative with this one. Anyway, I miss you. I hope you're all doing well. I'm going to eat this burnt marshmallow, and you guys have fun making some DNA. Maybe you can use marshmallows to make yours. All right, bye.